mitosis, meiosis, and then genetics, and then it's this new stuff that we're covering. So since it's a bunch of new stuff and it's right before the test, like basically learn it and then have an exam on it the next day, I'm gonna let you all have a card. You can put anything you want on it, front and back, that you can use. And what I've actually found is that when you make a card like this, you actually end up learning it a lot of times because you think about what is the most important thing to put on here. So everybody can take one of these and fill it with whatever it is that you have trouble remembering. Yeah, so that's, I think in the summertime especially, this is when I feel so bad about cramming your heads with all the knowledge and then you have the exam like the very next day. I also put up a study guide, similar to the one that I took last time that, again, these are just, I kind of went through my own PowerPoints and, on each slide, I kind of figured out what were we really focusing on. So that's, if you like to have that as a way to study, I still think reviewing the PowerPoints yourself and deciding which you think is important, rewriting your notes or, making your own cheat sheets, those are really the ways to move it to your long-term retention. Anybody have questions about any, like the last pieces of the quarter? So the research poster that's worth 50 points, so that's, that is half of an exam, and most of the time if people follow the rubric, they usually get the full points on that. I guess the things I would suggest just based on past quarters, make sure that you have for any figures, you have captions, for any gels, you have labels. Those are easy places where you can get points off, and it's easy to make sure you have those details. The other thing, what else? Oh, the other thing I was gonna say is the lab notebook. So the final lab notebook is due on Thursday, or I guess I said Friday. I will be going through it based on the rubric. It's kind of very similar to how you did your own self-assessment and you looked for different things. I kind of go through all the labs and look for, your purpose sections, your background sections, and just see if you consistently had thorough sections. So if you've had any notes along the way on your pre-labs, then it's a good time to make those changes. Yep.
Do we have to turn in anything for the lab?
No.
You just go in.
I just go in and look at it and then, yeah. So just make sure it's up to date by that point. Any other questions? Yeah, Tara, so this is you want to come and get that. This is for the final exam? Okay, good. You can use that so you can add those to it.
Oh, okay.
Okay, so I guess does anybody have any other questions? The other thing I noticed, are you able to do course evaluation still? Okay, so some people have submitted that and I give extra credit for that. So if you haven't done them, that's some extra credit points. You can easily do it and submit that. Okay, so I guess that is all I have. For review, I wanted to go back since a lot of the energy and enzymes you did on your own, you watched the video and then you filled out the ICA. But I noticed there was a little bit of confusion, so I want to review endergonic and exergonic. And to start with, I'm going to give each little group, I guess, one of these. I want you to decide, would this be an endergonic reaction or exergonic? And would it require or release energy? I don't know, do you want to work with, or maybe Sarah. We're missing a few people.
And I think that's
exergonic and it requires energy.
yeah.
Because breaking down releases energy, right? Yeah. It takes energy to start it.
It goes into release glucose. No, I'm just trying to yeah. Yes.
So it's form of so it's releasing.
All right, so
Exergonic, yeah.
so you want to go around and you want to share what you have written on your sticky note, and then
The what?
if you've got endergonic or exergonic and
The
require or
enzymes,
resist energy.
Oh, okay. Sorry, I just had a heart attack.
Oh, you're fine.
Wait,
Okay, and
exergonic.
endergonic. Okay, so did everybody hear that one? Synthesis of something, synthesis of a nucleotide. So maybe I'll put these categories here: endergonic and exergonic. So endergonic synthesis of a nucleotide. Okay, and that was the keyword. All right, how about yours, Ken?
So we got the hydrolysis of peptide chain. So this is like breaking the bond, so the potential energy from the bond will release. So there's hexagon.
Okay, yeah. So this is hydrolysis of a peptide chain. Okay, and that explanation was good, that you're releasing it, you're breaking a bond, you're releasing energy. Okay, so how about in the far back?
We had generation of starch. We're going from little to big. We're creating bonds, so we're storing energy in those bonds. So it's
Okay.
endergonic.
Endergonic, yeah. So generation is a keyword. Generation of starch from glucose. Okay, how about yours?
Ours is breakdown of glycogen to release glucose. So it's going to be exergonic.
Yeah.
Mhm.
What did it say, breakdown?
Yeah,
Yeah,
breakdown
breakdown.
of glycogen to release glucose.
Okay, so breakdown of use glycogen, reduce glucose. So we're going from something that has many bonds to something smaller, and so we're releasing the energy. How about the last one?
We had conversion of glucose to glycogen,
Uh-huh.
and we said it was endergonic.
Okay, yeah. Good. So conversion of glucose to glycogen. Okay, and you said that this was endergonic because we're going from small, we're at creating bonds, and so we need to input the energy to create the bonds. All right, so you have a solid understanding of how those break down. Now what I want you to do is, one of the questions you had on the ICA was to draw an energy diagram where you plot potential energy, or just basically Gibbs free energy, with reaction progress. And in this reaction, they had said you're starting with glycogen, and then you are breaking it down into glucose, and then you are adding a phosphate to glucose to get glucose 6-phosphate. This is actually one of the first steps that happens in glycolysis, is a phosphorylation step. So I noticed some of you had it correct, but then for some there was some confusion. So what I would do, maybe with the person next to you, you can sketch it out on the back of your sticky note if you want to, but it takes them stepwise. So when you go from glycogen to glucose, you have to decide, was that endergonic or exergonic? And then as a separate reaction, think, when you go from glucose to glucose 6-phosphate, is that exergonic or endergonic? And you draw, you're gonna have two activation peaks. And you have to decide, is one endergonic, one exergonic, or, and then, what does that overall curve look like? So.
Wait, I'm trying to find my right. Okay, there you go. I got that one. I got a mistake in that one. I don't. It's like I understand, but I don't understand. She said that the glucose should be lower than the glucose 6-phosphate. I don't. Why?
Yeah, you're right.
That's how I drew mine. So I guess it goes lower than the.
Sorry, the glucose should be lower than 6-phosphate.
Yeah.
No, yeah, sorry.
So should it be endergonic?
Okay, I don't think I drew answer it correctly, but I did like.
So I guess
go-
then
you go
glucose 6-phosphate isn't endergonic, or is
Yeah,
it? Not-
yeah, you gotta like.
So I need like
yeah, 'cause
glucose
I
to
think
glucose
you're just showing the
6-phosphate.
activation
Endergonic?
like on the second slide, but it's gotta be like up and then up and then. Okay.
I'm trying to think when things happening.
You're fine.
So, both of those
I mean,
directions
I
are exergonic.
guess I understand it.
So let's,
So they're both
let's talk about the
exergonic, but
first
I don't
one.
understand
I
why.
know this is kind of small here on the board, but
For breakdown of
this first going
really
from
good
glycogen to
glucose,
glucose, how did we classify
So
glycogen
it's
to
like that.
glucose when we did it
Right,
exergonic?
it's exergonic, but it
Yeah,
requires
so
energy.
it was
So
here.
it should have
So when we show
like an
exergonic,
activation,
which one would
and
be
then
at
it
a
should
higher
end
potential
higher
energy?
than beginning.
Mm.
Glycogen, right? So we're gonna start, I like to just start by
I think
drawing.
what you, glucose needs to be just above the starch.
About
Yeah.
Start by drawing kind of just rough estimates of the energy level for each of these components. So glycogen is pretty high, glucose has a lower potential energy. And then if we had to kind of
I don't
put
know.
glucose 6-phosphate, looking at glucose to going to glucose 6-phosphate, which one has a higher potential energy between these two?
Glucose.
Yeah, and so we have to relatively place that. Is it gonna be, would you think, halfway in between, or is it just gonna be closer to one or the other? Either to glucose or to glycogen? We're adding one phosphate group, right? So we have one bond. So I would say it's pretty close to glucose, but it is higher potential energy. We're raising the potential energy by adding the phosphate group. So those are kind of the three relative positions for those different, substances, and then we have to add the activation energy for each, right? So if we look at our first reaction, glycogen to glucose, exergonic, we add an activation energy peak, because there is an input of energy needed for this. So if we were to show where's the activation energy, it's from the energy of the reactant to the peak energy that we have. When we go from glucose to glucose 6-phosphate, we're still gonna have a peak again. Now we're going from where the glucose is all the way to the peak of the activation energy hump. And so this is a greater activation energy, which makes sense because this is endergonic and this is exergonic. Overall, would you say that this is an endergonic or exergonic, the whole thing? The net reaction. You look at your beginning, your reactants and your products, which one would you think it would be?
Exergonic.
Still exergonic overall, even though there was an endergonic step.
Mm-hmm.
But we have a huge exergonic step at the start. So overall, this entire reaction is gonna be still exergonic, even though there is a portion of it that it does require an input of energy. So does that make sense to everybody? A few of you had that right, but I noticed that there was some confusion about that. Okay, so if, like on a quiz, for example, one thing I might show you is, or give you a scenario like this and you have to classify it as endergonic or exergonic. Or if I just drew an energy diagram like this, could you classify it as being endergonic or exergonic just by looking at the energy diagram? Does everybody feel comfortable with that?
Mm-hmm.
Would that one be
Endergonic.
endergonic?
Yeah, this one would be endergonic. And the other thing that you should be familiar with is knowing, where is the activation energy on this? It goes from the energy of the reactants all the way to the peak. So activation energy here is pretty big. If we had an exergonic like this is the activation energy. Sometimes students will label like this whole peak and say that's the activation energy, but it's just the difference between the reactant and then the peak of the activation energy. The other thing is to know which one of these would be spontaneous, which one would occur spontaneously. Exergonic ones. So if delta G, right, if we have a negative delta G, that's a spontaneous reaction. that's looking at the change in the energy from the product to the reactant to the product. And then here, if it's positive, that is not spontaneous. That takes an input of energy. Okay, everybody feel comfortable with those types of questions? Okay. All right, so now we're gonna shift gears. Let me share screen on this Zoom. All right, so move this out of the way. Okay, so we are going to be doing, talking about cell respiration before. I know you've all learned about this in Bio 160. That's one of the units that I know is covered. We go into a little bit more detail for the steps. So you will be expected to know it at a slightly deeper level than what we did for Biology 160. But the other thing I like to do is give context for how does this fit in with what, with diet. And obviously, when we're looking at cell respiration, we're looking at how we harvest energy from things that we take into our body. So we'll talk about how all of this fits into the different macro molecules that we do consume in our diet. And luckily for you, since it's summer, I don't make you memorize all the parts of the pathways like I do in the normal course. But this diagram right here is a very complex diagram to give you an idea of how complicated all of these metabolic pathways really are. And if you go on, I know a lot of you want to go to medical school or plan to take biochemistry. And when you take biochemistry, you end up learning about all of these different metabolic pathways. And so you in detail, you learn how nucleotides are synthesized. And you learn how lipids are built, like fatty acids and glycerol. All of this, you learn how all of this is built. For us, we get to focus on this central metabolism pathway. So central metabolism is looking at carbohydrate metabolism, essentially how we take glucose and we break it down to produce energy. What's important to note though, what I want you to see is each of these dots is a intermediate. So it's a chemical compound along the way. And what you notice is that these are connected to all of the other metabolic pathways. So the purple one right here, you see this connects to amino acid metabolism, nucleotide metabolism. This one right here connects to lipid, even to carbohydrates. So this, I'm going to kind of tie this in, is that when we think about central metabolism, a lot of times we think about, we need to make energy, but really this has another purpose, and that is to create precursors for all of these other metabolic pathways. So far, you've learned about why do we do cell respiration, and the explanation you've probably already heard many times is that we need to make ATP, right? When you talked about what does the mitochondria do, you learned we need to make ATP. So that is one purpose for this, and this kind of shows you the very general scheme here. We're going to start with glucose, convert it to pyruvate through the process of glycolysis, and then pyruvate gets oxidized to produce acetyl-CoA, which can go into the citric acid cycle. After we've gone through the citric acid cycle, there's no more carbon left from the original glucose, but what we have created, anybody know what you create along the way that feeds into the electron transport chain? This might be stretching back into your minds.
Oxygen.
Well, we're going to use oxygen, yes. Oxygen will be the acceptor. Yeah. But anybody remember what the electron carriers that are made are called? NADH. Yeah, so we make NADH along the way, and we make FADH too. Those are going to feed into the electron transport chain, and then, as we mentioned, oxygen is going to be the final acceptor, and we're going to produce energy in that process. So that's one reason to do cell respiration. The second reason that I tried to show you on that previous slide is that it also is important for making biological precursors. So if you want to synthesize nucleotides for DNA replication or transcription, right, you you actually use some of these components. And this shows you here a much more complicated picture, but it shows you how each of these components can be used for other synthesis of other macromolecules. So you can make glycogen or starch as storage molecules from glucose. Acetyl-CoA is a really key one that is used to make fatty acids. And fatty acids, of course, are found in triglycerides or fats, and our membrane, our phospholipids. The citric acid cycle is used to make many of our amino acids. Now, we can't synthesize all of our amino acids, but the ones that we are able to make, most of them come from here. So this just adds in a level of complexity. Above, you kind of see how all of our macro nutrients that we consume, how they feed into this. We don't just eat glucose, right? That's not your diet, is not pure glucose all the time. In fact, a lot of people try to cut down on carbohydrates. We can talk a little bit about like what are the effects if you switch to a diet that reduces carb intake. But if you're eating carbs, this is how they're gonna enter in. They're gonna be converted into glucose or other sugars along the way in glycolysis. Your fats and phospholipids, they typically feed in at this acetyl-CoA level. Glycerol can be converted ultimately into pyruvate. And then proteins, they can either feed in, some of them feed in earlier steps. They can be converted to pyruvate, and others, if you deaminate them, they become intermediates in the citric acid cycle. So all of our foods that we consume somehow get into this metabolic pathway. Anybody have any questions so far? Okay, so to kind of lay out the stages, we will talk about 4 main stages of cell respiration. So that's glycolysis, which I know you probably have learned a overview of all of these stages. Pyruvate oxidation, you can put in your notes, that's also called the preparatory reaction. I know that the other textbook called it preparatory reaction. I can't think of other words for it, but our textbook calls it pyruvate oxidation. So that's going to take that pyruvate that comes out of glycolysis and makes it into acetyl-CoA, which then goes into the Krebs cycle. The third stage has lots of names, as you've seen already. It's called Krebs cycle, citric acid cycle, TCA cycle. Those are all talking about the same thing. So that's why I put them all there, just so that you, if anybody says a different term, you still know what we're talking about. And then the last stage is, of course, electron transport chain. If we have oxygen as a final acceptor, then that is called aerobic respiration. So most of what we do, right, in our cell respiration pathway is that we use oxygen as our final acceptor. There are other organisms, though, that don't live in an environment with oxygen. So there's some microbes that live in an anaerobic environment. They can still do these 4 steps. They just can't use oxygen as a final acceptor. So there's also anaerobic respiration. But what the nice thing here is that you're still getting all of the ATP production that we can get by going through the 4 stages of cell respiration. So we'll talk about what are some alternative acceptors. They can use other things other than oxygen as the final acceptor. And then one other part of this is that for organisms that do aerobic respiration, sometimes they don't have oxygen available. So if you think about exercise, when you first start running or something, in the very beginning of it, immediately your heart doesn't increase its rate of pumping, right? It takes a little bit of time for it to increase the rate of pumping. In that time, you might not be able to get enough oxygen to your muscles to be doing these 4 stages. So in that time, anybody know what you do instead in the absence of oxygen?
Create like lactic acid.
Yeah. Yes, so that's called fermentation. There's lactic acid fermentation, which is what we do. Other organisms will do ethanol fermentation, right? They create ethanol as a byproduct. So if there is no terminal acceptor, temporarily you can go through glycolysis, fermentation, and you make a little bit of ATP until your heart rate can increase to start delivering oxygen. Okay, So let's talk about ATP, since that is one of the things, the goal of these pathways is to create ATP. There's two ways that you can create ATP, and I'm sure you've kind of learned about these. Sorry, you can't see the top there. You probably learned about these different ways of making ATP in other classes. So the first method of making ATP is called substrate level phosphorylation. In your notes, you can write that this is the method that we use during glycolysis and during the Krebs cycle. So when we're making ATP, substrate level, we do this during glycolysis and Krebs cycle. What this method does is it uses a high energy phosphate bond. It breaks a high energy phosphate bond, and that phosphate is transferred to ADP, adenosine diphosphate, to create ATP. This is an enzyme catalyzed reaction. So this is going to be happening by enzymes in the glycolysis pathway or Krebs, in the Krebs cycle. And for each of these substrates, one of the things that you have to know is that if we're going to break this bond and add it to ADP to create this high energy ATP, this bond has to actually have more energy than the bond that we're forming. So these require, for substrate level phosphorylation to happen, you have to have a really high energy phosphate bond. So this, I took this from a paper. Don't worry about most of the details here. I'm not going to ask that you know these numbers. I just want to highlight the numbers we've already talked about. So 7.3, does that number ring a bell? Hopefully you remember that we talked about when you hydrolyze ATP, you get 7.3 kilocalories per mole. If I want to make ATP, I have to have an energy higher than that, greater than that, to be able to produce ATP. So when we get to glycolysis and we talk about what are the intermediates, what are the phosphorylated intermediates that can make ATP, you will find that they are all up here. Phosphoenolpyruvate, or PEP, we're going to come back to that one. That has a higher energy. And then this one, 1,3-bisphosphoglycerate. Those two are used to make ATP because the amount of energy they hold in their phosphate bond is higher than the amount of energy that we need to produce ATP. Now, this one I like to point out, how many of you have heard of phosphocreatine before? Usually people who are like in the gym or they're really into working out have heard of phosphocreatine.
That's like one of the other ways to get ATP, right?
Yeah, exactly. So if we have our kind of store of ATP, we use that up actually very quickly. We only have enough for like 3 seconds worth of activity, not very much. We can regenerate about 10 seconds worth of ATP by using phosphocreatine. So phosphocreatine carries the phosphate and can transfer it to ADP to make ATP. A lot of times athletes, or I won't say a lot of times, but some athletes will take phosphocreatine. And what's interesting, I noticed that there's sort of like false advertising about phosphocreatine because what it really can do is to increase your quick starts, like your sprint starts. It's not gonna build muscle or something, and I've seen advertising that kind of suggests that it can be taken to somehow build muscle. But anyway, the point of this is you can replenish your ATP stores. You have to have a higher energy in order to make the ATP. Okay? So that would be substrate level phosphorylation. The second method is oxidative phosphorylation in cell respiration, and I guess you can add one here to the list when we get to photosynthesis, and that is photophosphorylation. So photophosphorylation and oxidative phosphorylation are similar in that they both use an enzyme called ATP synthase, as well as an electrochemical gradient to produce ATP. The difference is for oxidative phosphorylation, which is used during cell respiration, the electrochemical gradient is produced as electrons move towards oxygen. I think that's why it's called oxidative, right? And then in photosynthesis, it's light that generates, that ultimately helps to create the electrochemical gradient. So that's why it's called photophosphorylation. But the result is the same. This proton gradient that is created, the protons move down their gradient through ATP synthase, and this spins like a motor and produces ATP. So those are the two types of ATP production. Does anybody have questions about those different types? Okay. So we're gonna go through these 4 stages, and this I took from, I think it came out of actually a nutrition textbook. Well, the reason I included this instead of from our textbook is that it sort of shows you kind of where things are taking place. So glycolysis is gonna happen at the beginning, and it shows you what our outputs are. So we're gonna create a little bit of ATP as well as electron carriers. This happens in the cytoplasm. So that's an important thing to note, is that glycolysis does not take place within the mitochondrion at all. And once you create the pyruvate, pyruvate has to be shuttled into the mitochondrion. Okay, so within the mitochondrion, the inner portion of the mitochondrion, actually we should kind of review the parts of the mitochondrion for a second before we move on. So hopefully you remember from chapter 3, we remember that there's 2 membranes, right, for mitochondrion. Does anybody remember what the space all the way in the middle was called? For that. Starts with an M. That matrix. Okay, so this is the mitochondrial matrix. The outer membrane is just called the outer membrane, inner membrane, and then there is this space in between that's called the intermembrane space, or the IMS, intermembrane space. So glycolysis takes place in the cytoplasm, and then pyruvate moves all the way into the matrix, so it has to go through 2 membranes to get into the matrix. And that is where pyruvate oxidation, or the preparatory reaction, that's where that happens. Still on the inside here, you have the Krebs cycle, or the citric acid cycle. And then you have all of the electron carriers that you need to feed into the ETC. Does anybody remember where the proteins of the electron transport chain are found? So they're going to be found in a membrane. Which membrane is it? Anybody remember from Bio 160? So all those proteins are found in this inner membrane. So you're going to hand off all of your electron carriers to those proteins here. And of course, the final production will be ATP and water, because the oxygen is going to be accepting electrons with some protons to create water. Okay. So for each of these stages, I try to give you kind of what is the takeaway for each stage. What do you need to know for each of the stages? So this is kind of in a nutshell what you should know for glycolysis. The first thing is, what's the purpose of the stage? What are we doing with the stage? And then where is it located? What are the inputs? What are the outputs? So the first one, what is the goal for the stage? The name itself kind of gives you a clue about this. So glyco is sugar. Lysis, we know, is breaking. So we're breaking down glucose, right? And we're going to produce 2 3-carbon sugars, which are called pyruvate. So we're going to go from glucose into 2 molecules of pyruvate. Where did we say the location was for this? In the cytoplasm. And so now we have to kind of put our overall inputs and outputs. So we're going to start from the perspective of 1 molecule of glucose. So if we put 1 molecule of glucose in, we are putting in 6 carbons. Anybody remember how much ATP is invested? This is drawing back to your memory of biology. Any guesses?
2.
2.
It's small, it's a small number. It's just 2. Okay, so we input 2. So you can put 1 here, 2 molecules of ATP. And then for NAD+, we also put in 2. So 2 molecules of NAD+. Remember with NAD+, we kind of talked about NAD as one of our electron carriers, and it has those two forms, NAD plus, NADH. Which form is the oxidized form? NAD plus. So this one is oxidized, it's ready to pick up electrons. This is with electrons, which is what gives us this hydrogen and the electrons there. So this one is the reduced form. So you put in 2 NAD plus so that it will be able to pick up electrons. And then the question is, do we have to have oxygen for this stage? So is this, you can say yes or no. Not required. Remember that before you increase your oxygen flow to your muscles, you can do glycolysis to produce a little bit of ATP. So no oxygen required.
You said how many molecules with NAD plus?
2. Yeah, 2. Then for the outputs, this is all from the perspective of that 1 molecule of glucose. We're going to get out, we're going to split it in half. So we get 2 molecules of 3 carbon. We're keeping track of where the carbons are going. So we still have all 6 so far. And for ATP, we're going to produce a total of 4 molecules of ATP, but we already used 2, so that's why we say the net is 2. So you can put 4 for this molecules of ATP, and then our net will be 2 produced from glycolysis. And since we put in 2 NAD+, we're going to get out 2 NADH. Those will go on. Where are they going to, where do the NADH end up?
The line.
Yep, in the electron transfer chain. So those will carry, they're kind of temporarily holding onto this energy that will later be harvested. Okay, so any questions on overall for glycolysis? Okay, so here's where you all are lucky. So normally during the school year, we have a little bit more time. Usually we require the 211 students to know the names of the intermediates and the names of the enzymes. So you don't, not that you have to draw them, but that you have to recognize them. But because it's short, we're in a quick quarter, I do want you to know the type of reaction. So for each one of these, there's 10 steps. What type of reaction is it? But you don't have to know inputs and outputs, and you don't have to know the enzymes. Okay? So let's talk about these. Now, what I like to focus on when you're learning types of reactions is kind of helps to know why each step is happening. And I think actually that's the bigger goal, even in biochemistry, than memorizing all these intermediates, because you can look that up. But knowing why every step is happening, that's probably more important to understanding the process. So for glycolysis, the first 5 steps are called the energy investment phase. This is where we're going to put in those 2 ATP. When we're putting ATP into something, we're going to be raising the potential energy. So just like I drew right here, we went from glucose 6-phosphate, we raise the potential energy by adding a phosphate. So phosphorylation has a purpose. Later on, you can get that energy back out, but it's kind of a temporary hold on the energy. So let's look at the steps. I'll kind of walk you through what's happening. You won't have to recognize these. So glucose is the input. The very first step is going to use ATP, and this enzyme called hexokinase is going to add the phosphate from ATP to glucose. Now we create glucose 6-phosphate, just like I had in this example right here. So this has raised the potential energy. What type of reaction would this be? You can put this in your notes over step 1. It's called a phosphorylation. Makes sense, right? We're adding a phosphate group. And the reason for that is we're trying to, actually a twofold reason. 1, it raises potential energy, and 2, it traps the glucose in the cell. So adding that phosphate keeps it inside the cell. Then we go from glucose 6-phosphate to fructose 6-phosphate. So glucose and fructose, you've learned about these as monosaccharides, right? They generally, if you count up the number of carbons, oxygens, hydrogens, it's exactly the same. But what has happened if you just look at the structures? Yeah. And then how many sides we have here? 6? 5. So this is just a rearrangement. Anytime you're rearranging, that's called an isomer isomerization. You probably remember from chemistry, isomers, so different chemical arrangements of the same atoms. So this is an isomerization reaction. We go from glucose 6-phosphate to fructose 6-phosphate. The next step, step 3, again requires ATP. So what type of reaction do you think it would be if we're using ATP? And we're adding a phosphate? Phosphorylation. Okay, so this is another phosphorylation. Now I went from fructose 6-phosphate, fructose 1,6-bisphosphate. I added another phosphate group. Now I've raised the energy up even higher. Ultimately, I'm going to collect that energy back out. The fourth step, you see what happens. This is important because this is where the pathway kind of bifurcates, and you split it into 2 molecules. So one is called dihydroxyacetone phosphate, or DHAP. The other one is glyceraldehyde 3-phosphate. This one, I want you to circle this. You will see this intermediate in photosynthesis when we're creating sugars, and you see it as a breakdown product in glycolysis when we're breaking down glucose. This is what is ultimately going to go on to the next step. So even though we have 2 different molecules, they both have to be converted into the same thing. They both have to be converted to G3P. So step number 4, which of these do you think would describe step number 4? Cleavage. We're cutting it in half. So step 4 is a cleavage. This DHAP converting it into G3P is just a rearrangement. So what's that called? Isomerization. Number step 5 is isomerization. Okay, so that is the first 5 steps of glycolysis are just basically getting everything primed and ready to release the energy. So then we go to the energy payoff phase. And from this, actually you should note that, remember that we converted to G3P. So all of this stuff happens in parallel. So I like to draw it out, I don't know, for myself, just to organize this. In my notes, I would have something like this, glucose, and at some point it splits off into 2 parallel pathways. Step 1 requires ATP, step 3 requires ATP. And then once you get here, these are both being converted into G3P. So all of this is happening in parallel. Everything that you see here, you can see they put the number 2 next to everything because everything's happening in parallel. So step 6 is adding a phosphate, but here's what's interesting about this step. It also adds NAD+. So whenever we're adding an electron acceptor, we're going to have a redox reaction. So this type, for step 6, you can say that step is a redox reaction. We're going to create NADH, which is holding on to some of the electron energy. So I would draw on my little schematic here what's coming off. Step 6 is I'm creating NADH twice, because it's happening on both of those molecules. Then we create 1,3-bisphosphoglycerate. Where does that sound familiar? I just showed you a chart with it, right? So this one, 1,3-bisphosphoglycerate, has a very high, this phosphate bond right here, is a very high energy phosphate bond. So I can break that, and it has enough energy produced to make ATP. So then I add this phosphate to ADP. This is my first production of ATP. So step 7, what would you think that is of these 4? If you're producing, you're moving a phosphate group. Yeah, it's a phosphorylation. And this time we're making ATP. So step 7, you can make ATP, you're doing it twice.
What was the reaction for the first part of this?
6,
Yeah.
redox. This one called redox is called redox because of the NAD+ that's going to accept the electron. Okay, so now I've gotten rid of this phosphate. I added it to ADP. Now I create 3-phosphoglycerate. The next step, I go from 3 to 2. All I did was I moved this in a different position. So what type of reaction would that be? You're just moving a phosphate. Isomerization. So this one's isomerization. The reason I was doing that, I'm getting it into the right position to ultimately I want to produce phosphoenol pyruvate. That's the other one I told you about, PEP. This is going to be another high energy phosphate bond. So I have to actually, in order to do this, I need to create this high energy intermediate, which is an enol. Enol is a double bond with an alcohol. So that's the higher energy bond. To do that, I actually take out water. So this is one that's not listed here. It's a dehydration. And when I take out a molecule of water, it actually creates this high energy phosphoenol pyruvate. So step 9, you can write is dehydration. And then I have PEP. And PEP has that high energy phosphate. So again, I bring in ADP and I can make ATP. So what type of reaction is that? Creating, phosphorylation. Last step creates pyruvate. So step 7 and step 10 are the 2 places where we are making ATP. If you count them up here, we have 4 ATP produced. You have 2 NADH. And then we used minus 2 ATP. So that was used in the beginning. At the end of this, what we created was pyruvate from this one and pyruvate from this one. Okay, and keeping track of carbons, I never lost any carbons. I started with 6. I still have 6. They're just in the form of pyruvate right now. All right, any questions on those steps?
I don't know if this is a silly question or not, but is the creation of pyruvate why we get energy boosts from eating sugar?
Is the creation of pyruvate, so our sugar is going to feed through this glycolysis and then into the TCA cycle, and pyruvate is important to then be able to create acetyl-CoA. So I don't know if that answers your question. So you're saying, is that why we create sugar? Or why we create energy?
Yeah, like when we eat sugar, we get like an energy boost, like a sugar high.
Is it that,
Yeah.
like, false, or is it that, like, a I don't know what word is it. We think that you get sugar rushes, but you don't actually. I mean,
Like
like,
a
just with
sugar-
energy,
a
but, no.
Yeah, so I guess
Okay.
the pyruvate itself, I don't think explains a sugar rush. I don't know if you're thinking of like
I
a
mean-
spike in your blood glucose levels. I don't know
Yeah, I don't
any
know.
more about with the Mythbusters, for that. But pyruvate itself shouldn't be giving like a sugar rush, ultimately.
Like the creation of the 2 ATP. No.
Oh, the 2 ATP.
Yeah.
I don't think that's gonna give, I don't think that gives you a sugar rush. Because the energy boost. Yeah. Anyway, we can look more into it. Okay, so this I included because, like I said, the important part of understanding glycolysis is kind of understanding why things are happening. So I explained it verbally, but if you want to come back and review, this explains why every step happens. This tells you why do you need to transfer a phosphate group? Why do you need to transfer electrons to NADH? All of this, or to produce NADH. So this gives you the reasoning behind each of these. You don't have to know the enzymes over there, but those are the enzymes that catalyze each of those 10 reactions. Now, this is really a biosynthetic, this is a metabolic pathway, right? So if we're gonna have a metabolic pathway, we're trying to create ATP. We need to have a way to sense when is enough. Like, have we made enough ATP? When should we shut down this reaction? So in order to do this, we have a way to feed back. We have a way to sense, do we have enough ATP? Should we keep going through glycolysis or should we stop? And so most of these metabolic pathways have feedback regulation. In this case, we're gonna regulate an early step in the pathway. Why would it be better to regulate one of the earliest enzymes instead of regulating one of the later enzymes?
So you only.
Yeah, exactly. So with these types of feedback mechanisms, you're going to have typically a product of the reaction often will feed back at an early step in the pathway and inhibit one of the early enzymes. The enzyme that is regulated for glycolysis is called phosphofructokinase, or PFK. This is catalyzing the third reaction. And the reason that we regulate this step is because this is a committed step in glycolysis, which means once you go through this step, you're gonna go through all of the 10 steps of glycolysis. The first 2 steps are reversible. So you don't have, if you start it, you can actually go backwards in those 2, and you're not committed to going through all of the 10 steps. So it makes sense, this one is gonna be a regulated step. If we look at what PFK does here, it's taking fructose 6-phosphate and converting it to fructose 1,6-bisphosphate. So it has to bind this, and it has to bind ATP in order to catalyze this reaction, and then it's gonna transfer one of the phosphates to fructose 6-phosphate to create that fructose 1,6-bisphosphate. So the key here is that it already binds to ATP. And ATP is kind of the product of all of this, right? We're trying to make ATP. So the key to regulation is that there's actually 2 different ATP binding sites. This is the active site, but there's also an allosteric. Remember learning about allosteric inhibitors? Allo means other, and steric is site. So there's another site on the enzyme that binds to ATP. When ATP binds here, it actually changes the conformation of the enzyme so that the active site is no longer able to catalyze the reaction. So the key here is that there are actually different affinities of these 2 sites. Because the enzyme has to be able to bind ATP, we don't want ATP to bind over here when it actually is supposed to be binding here. So the active site has high affinity for ATP. So that means that when the cell has low ATP levels, if there's a little bit of ATP, it's going to prefer to bind at the high affinity site that has stronger binding. However, if the cell is swimming with ATP, there's tons of ATP, then ATP can bind at the low affinity site. And that will happen more often, which means that this will change conformation and it will prevent this reaction from taking place. So if glycolysis is inhibited, glucose is going to build up, and then we can't just have a ton of glucose floating around. So what does glucose get made into? Glycogen. So then glucose would be converted into glycogen. If we've got plenty of ATP and that's the signaling that we need to go backwards, that's what will happen when glycolysis is inhibited. Okay, any questions on that, how PFK works for regulation here? Okay, so this is an example of negative inhibition, or negative feedback. Feedback inhibition is what I was thinking of. So phosphofructokinase is inhibited, as you can see here, inhibited by ATP. It also can be inhibited by a buildup of citrate. So another later step in the pathway, if this builds up, it will also inhibit the very early steps of the pathway. What stimulates phosphofructokinase is AMP, which is kind of a readout in the cell for low energy levels, for low energy state.
So the same AMP that gets turned into CAMP?
Yeah, or CAMP gets turned into AMP, yeah. Okay, here's a question to discuss with the person next to you. If we had phosphofructokinase mutated such that the allosteric ATP binding site can't bind to ATP, what would be the effect on ATP production in the cell? So we're saying that just the allosteric site is changed so that it doesn't recognize ATP. So what do you think would happen with ATP production?
Uninhibited.
Yeah, so they can't produce any more ATP. It's going to change the shape of it, isn't it?
No, we make too much ATP.
Oh, really?
The allosteric site is the one that
Oh, yeah.
ATP binds
right,
too
right,
much.
I see. Okay.
So it would never be inhibited. So it just make like
Mm-hmm.
ATP forever. And ever and ever and ever.
Okay, so I got it out. Okay.
Electrolysis is renewable.
Okay, so what would happen if it can't recognize ATP in that site? Still can recognize it in the active site. Any thoughts? Sure.
Well, there'd be a lot, like excessive ATP production, and then because of that, glucose would never get converted to glycogen.
Yeah, so we might expect an increase in ATP production. It's not the only kind of regulatory point, but we definitely would see a change because this is a key regulatory point. So probably you can put there something about increase in ATP production, maybe less glycogen produced, because we're not actually kind of listening to when we should switch off this and start making glycogen. Okay, so that's glycolysis. Now everybody we've gone through glycolysis so far. So I want everybody to take out your ICA. You should be able to fill out this row. And there's a couple things, for ATP you have to think about, was the ATP that was created, was that substrate level or was that oxidative phosphorylation? So you can put 0 if it was not produced in that, way. And then CO2, did we produce CO2 in this so far? Have we made any CO2? Not yet. So you put 0 for anything that we haven't yet made. Okay, just the outputs.
You want the output, like, if there's 4 different molecules that.
I would say the net, but you can put in parentheses, like 2 net or total or something, just so you also can study it, but